A tendon or sinew is a tough band of fibrous connective tissue that usually connects muscle to bone and is capable of withstanding tension. Tendons are similar to ligaments, both are made of collagen. Ligaments join one bone to another bone, while tendons connect muscle to bone. Structure. Histologically, tendons consist of dense regular connective tissue fascicles encased in dense irregular connective tissue sheaths. Normal healthy tendons are composed mostly of parallel arrays of collagen fibers closely packed together. They are anchored to bone by Sharpie's fibers. The dry mass of normal tendons, which makes up about 30% of their total mass, is composed of about 86% collagen, 2% elastin, 1.5% proteoglycans, and 0.2% inorganic components such as copper, manganese, and calcium. The collagen portion is made up of 97–98% type I collagen, with small amounts of other types of collagen. These include type II collagen in the cartilaginous zones, type III collagen in the reticulin fibers of the vascular walls, type IX collagen, type IV collagen in the basement membranes of the capillaries, type V collagen in the vascular walls, and type X collagen in the mineralized fibrocartilage near the interface with the bone. Collagen fibers coalesce into macroaggregates. After secretion from the cell, the cleaved by procollagen N and C proteinases, and the tropocollagen molecules spontaneously assemble into insoluble fibrils. A collagen molecule is about 300 nm long and 1–2 nm wide, and the diameter of the fibrils that are formed can range from 5,500 nm. In tendons, the fibrils then assemble further to form fascicles, which are about 10 mm in length with a diameter of 5,300 μm, and finally into a tendon fiber with a diameter of 100–500 μm. Fascicles are bound by the endotendinium, which is a delicate loose connective tissue containing thin collagen fibrils and elastic fibers. Groups of fascicles are bounded by the epitenon. Filling the interstitia within the fascia where the tendon is located is the paratenon, a fatty areolar tissue. The collagen in tendons are held together with proteoglycan components including decorin and, in compressed regions of tendon, aggrecan, which are capable of binding to the collagen fibrils at specific locations. The proteoglycans are interwoven with the collagen fibrils. Their glycosaminoglycan, GAG, side chains have multiple interactions with the surface of the fibrils, showing that the proteoglycans are important structurally in the interconnection of the fibrils. The major GAG components of the tendon are dermatan sulfate and chondroitin sulfate, which associate with collagen and are involved in the fibril assembly process during tendon development. Dermatan sulfate is thought to be responsible for forming associations between fibrils, while chondroitin sulfate is thought to be more involved with occupying volume between the fibrils to keep them separated and help withstand deformation. The dermatan sulfate side chains of decorin aggregate in solution, and this behavior can assist with the assembly of the collagen fibrils. When decorin molecules are bound to a collagen fibril, their dermatan sulfate chains may extend and associate with other dermatan sulfate chains on decorin that is bound to separate fibrils, therefore creating interfibrillar bridges and eventually causing parallel alignment of the fibrils. The tenocytes produce the collagen molecules, which aggregate end-to-end and side-to-side to produce collagen fibrils. Fibril bundles are organized to form fibers with the elongated tenocytes closely packed between them. There is a three-dimensional network of cell processes associated with collagen in the tendon. The cells communicate with each other through gap junctions, and this signaling gives them the ability to detect and respond to mechanical loading. Blood vessels may be visualized within the endotendon running parallel to collagen fibers, with occasional branching transverse anastomosis. The internal tendon bulk is thought to contain no nerve fibers, but the epitenon and paratenon contain nerve endings, while Golgi tendon organs are present at the junction between tendon and muscle. Tendon length varies in all major groups and from person to person. Tendon length is, in practice, the deciding factor regarding actual and potential muscle size. For example, all other relevant biological factors being equal, a man with a shorter tendons and a longer biceps muscle will have greater potential for muscle mass than a man with a longer tendon and a shorter muscle. Successful bodybuilders will generally have shorter tendons. Conversely, in sports requiring athletes to excel in actions such as running or jumping, it is beneficial to have longer than average Achilles tendon and a shorter calf muscle. Tendon length is determined by genetic predisposition, and has not been shown to either increase or decrease in response to environment, unlike muscles, which can be shortened by trauma, use imbalances, and a lack of recovery and stretching. Functions. Traditionally, tendons have been considered to be a mechanism by which muscles connect to bone, functioning to transmit forces. This connection allows tendons to passively modulate forces during locomotion, providing additional stability with no active work. However, over the past 2 decades, much research focused on the elastic properties of some tendons and their ability to function as springs. Not all tendons are required to perform the same functional role, with some predominantly positioning limbs, such as the fingers when writing, positional tendons, and others acting as springs to make locomotion more efficient, energy-storing tendons. Energy-storing tendons can store and recover energy at high efficiency. For example, during a human stride, the Achilles tendon stretches as the ankle joint dorsiflexes. During the last portion of the stride, as the foot plantar flexes, pointing the toes down, the stored elastic energy is released. Furthermore, because the tendon stretches, the muscle is able to function with less or even no change in length, allowing the muscle to generate greater force. The mechanical properties of the tendon are dependent on the collagen fiber diameter and orientation. The collagen fibrils are parallel to each other and closely packed, but show a wave-like appearance due to planar undulations, or crimps, on a scale of several micrometers. In tendons, the collagen fibers have some flexibility due to the absence of hydroxyproline and proline residues at specific locations in the amino acid sequence, which allows the formation of other conformations such as bends or internal loops in the triple helix and results in the development of crimps. The crimps in the collagen fibrils allow the tendons to have some flexibility as well as a low compressive stiffness. In addition, because the tendon is a multi-stranded structure made up of many partially independent fibrils and fascicles, it does not behave as a single rod, and this property also contributes to its flexibility. The proteoglycan components of tendons also are important to the mechanical properties. While the collagen fibrils allow tendons to resist tensile stress, the proteoglycans allow them to resist compressive stress. These molecules are very hydrophilic, meaning that they can absorb a large amount of water and therefore have a high swelling ratio. Since they are non-covalently bound to the fibrils, they may reversibly associate and dissociate so that the bridges between fibrils can be broken and reformed. This process may be involved in allowing the fibril to elongate and decrease in diameter under tension. However, the proteoglycans may also have a role in the tensile properties of tendon. The structure of tendon is effectively a fiber composite material, built as a series of hierarchical levels. At each level of the hierarchy, the collagen units are bound together by either collagen cross-links, or the proteoglycans, to create a structure highly resistant to tensile load. The elongation and the strain of the collagen fibrils alone have been shown to be much lower than the total elongation and strain of the entire tendon under the same amount of stress, demonstrating that the proteoglycan-rich matrix must also undergo deformation, and stiffening of the matrix occurs at high strain rates. This deformation of the non-collagenous matrix occurs at all levels of the tendon hierarchy, and by modulating the organization and structure of this matrix, the different mechanical properties required by different tendons can be achieved. Energy-storing tendons have been shown to utilize significant amounts of sliding between fascicles to enable the high strain characteristics they require, whilst positional tendons rely more heavily on sliding between collagen fibers and fibrils. However, recent data suggests that energy-storing tendons may also contain fascicles which are twisted, or helical, in nature, an arrangement that would be highly beneficial for providing the spring-like behavior required in these tendons. Mechanics. Tendons are viscoelastic structures, which means they exhibit both elastic and viscous behavior. When stretched, tendons exhibit typical soft tissue behavior. The force extension, or stress-strain curve starts with a very low stiffness region, as the crimp structure straightens and the collagen fibers align, suggesting negative Poisson's ratio in the fibers of the tendon. More recently, tests carried out in vivo, through MRI, and ex vivo, through mechanical testing of various cadaveric tendon tissue, have shown that healthy tendons are highly anisotropic and exhibit a negative Poisson's ratio, auxetic, in some planes when stretched up to 2% along their length, i.e. within their normal range of motion. After this toe region, the structure becomes significantly stiffer, and has a linear stress-strain curve until it begins to fail. The mechanical properties of tendons vary widely, as they are matched to the functional requirements of the tendon. The energy-storing tendons tend to be more elastic, or less stiff, so they can more easily store energy, whilst the stiffer positional tendons tend to be a little more viscoelastic, and less elastic, so they can provide finer control of movement. A typical energy-storing tendon will fail at around 12 to 15% strain, and a stress in the region of 100 to 150 MPa, although some tendons are notably more extensible than this, for example the superficial digital flexor in the horse, which stretches in excess of 20% when galloping. Positional tendons can fail at strains as low as 6 to 8%, but can have moduli in the region of 700–1000 MPa. Several studies have demonstrated that tendons respond to changes in mechanical loading with growth and remodeling processes, much like bones. In particular, a study showed that disuse of the Achilles tendon in rats resulted in a decrease in the average thickness of the collagen fiber bundles comprising the tendon. In humans, an experiment in which people were subjected to a simulated microgravity environment found that tendon stiffness decreased significantly, even when subjects were required to perform restiveness exercises. These effects have implications in areas ranging from treatment of bedridden patients to the design of more effective exercises for astronauts. Healing. The tendons in the foot are highly complex and intricate. Therefore, the healing process for a broken tendon is long and painful. Most people who do not receive medical attention within the first 48 hours of the injury will suffer from severe swelling, pain, and a burning sensation where the injury occurred. It was believed that tendons could not undergo matrix turnover and that tenocytes were not capable of repair. However, it has since been shown that, throughout the lifetime of a person, tenocytes in the tendon actively synthesize matrix components as well as enzymes such as matrix metalloproteinases, MMPs, can degrade the matrix. Tendons are capable of healing and recovering from injuries in a process that is controlled by the tenocytes and their surrounding extracellular matrix. The three main stages of tendon healing are inflammation, repair or proliferation, and remodeling, which can be further divided into consolidation and maturation. These stages can overlap with each other. In the first stage, inflammatory cells such as neutrophils are recruited to the injury site, along with erythrocytes. Monocytes and macrophages are recruited within the first 24 hours, and phagocytosis of necrotic materials at the injury site occurs. After the release of vasoactive and chemotactic factors, angiogenesis and the proliferation of tenocytes are initiated. Tenocytes then move into the site and start to synthesize collagen III. After a few days, the repair or proliferation stage begins. In this stage, the tenocytes are involved in the synthesis of large amounts of collagen and proteoglycans at the site of injury, and the levels of GAG and water are high. After about 6 weeks, the remodeling stage begins. The first part of this stage is consolidation, which lasts from about 6 to 10 weeks after the injury. During this time, the synthesis of collagen and GAGs is decreased, and the cellularity is also decreased as the tissue becomes more fibrous as a result of increased production of collagen I, and the fibrils become aligned in the direction of mechanical stress. The final maturation stage occurs after 10 weeks, and during this time there is an increase in cross-linking of the collagen fibrils, which causes the tissue to become stiffer. Gradually, over about 1 year, the tissue will turn from fibrous to scar-like. Matrix metalloproteinases, MMPs, have a very important role in the degradation and remodeling of the ECM during the healing process after a tendon injury. Certain MMPs including MMP1, MMP2, MMP8, MMP13, and MMP14 have collagenase activity, meaning that, unlike many other enzymes, they are capable of degrading collagen I fibrils. The degradation of the collagen fibrils by MMP1 along with the presence of denatured collagen are factors that are believed to cause weakening of the tendon ECM and an increase in the potential for another rupture to occur. In response to repeated mechanical loading or injury, cytokines may be released by tenocytes and can induce the release of MMPs, causing degradation of the ECM and leading to recurring injury and chronic tendinopathies. A variety of other molecules are involved in tendon repair and regeneration. There are 5 growth factors that have been shown to be significantly upregulated and active during tendon healing: insulin-like growth factor 1, IGF-1, platelet-derived growth factor, PDGF, vascular endothelial growth factor, VEGF, basic fibroblast growth factor, BFGF, and transforming growth factor beta, TGF-beta. These growth factors all have different roles during the healing process. IGF-1 increases collagen and proteoglycan production during the first stage of inflammation, and PDGF is also present during the early stages after injury and promotes the synthesis of other growth factors along with the synthesis of DNA and the proliferation of tendon cells. The 3 isoforms of TGF-beta, TGF-beta1, TGF-beta2, TGF-beta3, are known to play a role in wound healing and scar formation. VEGF is well known to promote angiogenesis and to induce endothelial cell proliferation and migration, and VEGF mRNA has been shown to be expressed at the site of tendon injuries along with collagen I mRNA. Bone morphogenetic proteins, BMPs, are a subgroup of TGF-beta superfamily that can induce bone and cartilage formation as well as tissue differentiation, and BMP12 specifically has been shown to influence formation and differentiation of tendon tissue and to promote fibrogenesis. Effects of activity on healing. In animal models, extensive studies have been conducted to investigate the effects of mechanical strain in the form of activity level on tendon injury and healing. While stretching can disrupt healing during the initial inflammatory phase, it has been shown that controlled movement of the tendons after about 1 week following an acute injury can help to promote the synthesis of collagen by the tenocytes, leading to increased tensile strength and diameter of the healed tendons and fewer adhesions than tendons that are immobilized. In chronic tendon injuries, mechanical loading has also been shown to stimulate fibroblast proliferation and collagen synthesis along with collagen realignment, all of which promote repair and remodeling. To further support the theory that movement and activity assist in tendon healing, it has been shown that immobilization of the tendons after injury often has a negative effect on healing. In rabbits, collagen fascicles that are immobilized have shown decreased tensile strength, and immobilization also results in lower amounts of water, proteoglycans, and collagen cross-links in the tendons. Several mechanotransduction mechanisms have been proposed as reasons for the response of tenocytes to mechanical force that enable them to alter their gene expression, protein synthesis, and cell phenotype, and eventually cause changes in tendon structure. A major factor is mechanical deformation of the extracellular matrix, which can affect the actin cytoskeleton and therefore affect cell shape, motility, and function. Mechanical forces can be transmitted by focal adhesion sites, EYNTK grins, and cell-cell junctions. Changes in the actin cytoskeleton can activate EYNTK grins, which mediate outside-in and inside-out signaling between the cell and the matrix. G proteins, which induce intracellular signaling cascades, may also be important, and ion channels are activated by stretching to allow ions such as calcium, sodium, or potassium to enter the cell. Society and culture. Sinew was widely used throughout pre-industrial eras as a tough, durable fiber. Some specific uses include using sinew as thread for sewing, attaching feathers to arrows, see fletch, lashing tool blades to shafts, etc. It is also recommended in survival guides as a material from which strong cordage can be made for items like traps or living structures. Tendon must be treated in specific ways to function usefully for these purposes. Inuit and other circumpolar people utilized sinew as the only cordage for all domestic purposes due to the lack of other suitable fiber sources in their ecological habitats. The elastic properties of particular sinews were also used in composite recurved bows favored by the steppe nomads of Eurasia. The first stone-throwing artillery also used the elastic properties of sinew. Sinew makes for an excellent cordage material for three reasons: it is extremely strong, it contains natural glues, and it shrinks as it dries, doing away with the need for knots. Culinary uses. Tendon, in particular, beef tendon, is used as a food in some Asian cuisines, often served at yum cha or dim sum restaurants. One popular dish is swan bao niu jin, in which the tendon is marinated in garlic. It is also sometimes found in the Vietnamese noodle dish ph. Clinical significance. Injury. Tendons are subject to many types of injuries. There are various forms of tendinopathies or tendon injuries due to overuse. These types of injuries generally result in inflammation and degeneration or weakening of the tendons, which may eventually lead to tendon rupture. Tendinopathies can be caused by a number of factors relating to the tendon extracellular matrix, ECM, and their classification has been difficult because their symptoms and histopathology often are similar. The first category of tendinopathy is paratendinitis, which refers to inflammation of the paratenon, or paratendinous sheet located between the tendon and its sheath. Tendinosis refers to non-inflammatory injury to the tendon at the cellular level. The degradation is caused by damage to collagen, cells, and the vascular components of the tendon, and is known to lead to rupture. Observations of tendons that have undergone spontaneous rupture have shown the presence of collagen fibrils that are not in the correct parallel orientation or are not uniform in length or diameter, along with rounded tenocytes, other cell abnormalities, and the ingrowth of blood vessels. Other forms of tendinosis that have not led to rupture have also shown the degeneration, disorientation, and thinning of the collagen fibrils, along with an increase in the amount of glycosaminoglycans between the fibrils. The third is paratendinitis with tendinosis, in which combinations of paratenon inflammation and tendon degeneration are both present. The last is tendinitis, which refers to degeneration with inflammation of the tendon as well as vascular disruption. Tendinopathies may be caused by several intrinsic factors including age, body weight, and nutrition. The extrinsic factors are often related to sports and include excessive forces or loading, poor training techniques, and environmental conditions. Other animals. In some organisms, notable ones being birds and ornithischian dinosaurs, portions of the tendon can become ossified. In this process, Osteocytes infiltrate the tendon and lay down bone as they would in sesamoid bone such as the patella. In birds, tendon ossification primarily occurs in the hind limb, while in ornithischian dinosaurs, ossified axial muscle tendons form a latticework along the neural and hemal spines on the tail, presumably for support.
Please subscribe and thanks for watching.